Hello, and welcome to the last FanBug seminar of the 2023 to 2024 series. My name is Jonathan, and I will be your host for today. As a reminder, please silence your phones. This event is being recorded and will be uploaded to YouTube so that anyone who couldn't make it today will be able to check out the seminar. This is the agenda for today. I'll first finish the rest of the announcements, then we will hear from our introductory trainee, followed by a featured talk. And as always, there will be free pizza and some social networking, with the event ending at around 1:30 p.m. FanBug is a volunteer-driven organization, so we're very lucky to have our free faculty members, Dr. William Shao, Dr. Amy Lee, and Dr. Faraz Hush in helping us organizing these seminars. I'd also like to thank our student volunteers for their contributions in helping organizing this year's seminars. We're actively preparing for next year's seminars, so we welcome any suggestion for international and local speakers that you're interested in hearing from. And you can do that by going to our website and use our suggested speaker form and tell us who you'd like to listen from. We'd also like to invite you to subscribe to our mailing list for any updates, and you can do so by sending an email with a subject line "subscribe" to this following mailing address. You can also use this QR code to subscribe to our calendar. FanBug is sponsored by a number of organizations, including the Graduate Studies in Bioinformatics at UBC and SFU, Genome British Columbia. We're also grateful to the Big Data Hub at SFU and the Michael Smith Laboratories at UBC for allowing us to use their venues and hosting us this year. We're also sponsored by Langara and on the industry side, Stem Cell Technologies. FanBug is affiliated with our two sister bug groups, MomBug and TorBug, and we're also affiliated with the International Society for Computational Biology. As a note to the Zoom audience, please ask your questions in the chat and a FanBug volunteer will voice it for you. For questions coming from the other venue at SFU, we ask you reach out to the volunteers there and they will assist you in asking your questions. First, we will hear from our introductory student speaker, Jordan. He's a PhD student in bioinformatics at TAM and Frango Lab at UBC. And his talk is titled Brain Age Prediction, Systematic Evaluation of Site Effects and Sample Age Range and Size. We will then have our featured talk from Dr. Sabrina Leslie. She is an associate professor at the Michael Smith Labs and Physics Department at UBC, and she's also the Chief Scientific Officer and President of ScopeSys. And her presentation is on multi-scale seeing is believing platform and investigations, imaging and advancing RNA vaccines and genetic medicines. Now I'd like to welcome our student speaker to present. Yeah, use this.
So good morning, everyone. My name is Jordan, and I'm currently a PhD candidate at UBC Bioinformatics, supervised by Dr. Sophia Frangu and Dr. Roger Tam. and my research is focused on computational psychiatry. So today, I'm very excited to be here to share my most recent research about brain age prediction, systematic evaluation of side effects, and sample age range and size. So let's start by understanding what brain age really means. Essentially, brain age refers to the biological age of our brains, which might be different from our chronological age, and that's simply the number of birthdays we celebrated. Brain age has gained a lot of attention as a valuable biomarker because it helps quantify whether our brain is aging faster or slower compared to our actual age. And the difference between our brain's predicted age and our chronological age is what we call the brain age gap estimate, or brain age with capital letters of AGE. And this concept is crucial in studying how our brains age and how this aging is associated with many neurological and psychiatric disorders. And therefore, the need to develop accurate and robust brain age models is increasingly important. And there are actually several methods to predict the brain age, included by using genetic data, neuroimaging data, and so on. But for this study, we're focusing on using structural MRI data, which is one of the most commonly used type of neuroimaging used to assess the brain age. And in our previous study, we conducted a systematic evaluation of 21 algorithms to assess their performance, effectiveness, and efficiency in predicting brain age. And we found that the support vector regression with radial basis kernel, function kernel, has the best performance. So we've chosen this algorithm for our models. And our discovery sample is quite huge, containing structural MRI scans from over 35,000 healthy individuals, ranging in age from 5 to 90 years old, with approximately 54% of them are female. And this wide age range covers most of the human lifespan. And this provides a substantial basis for our analysis. The test set includes scans from over 2,100 healthy individuals. So with such a large and diverse data set, it might seem that more data automatically translates to a better model. But is that really the case, or are there any other factors might influence the performance of the model? So to address these questions, we've set three key objectives for our study. The first is to assess the effects of sample age range and site harmonization on the performance of machine learning models for brain age prediction. The second is to establish a minimum sample size requirement for getting a stable brain age model. And last is to test the longitudinal consistency of pre-trained brain age models. So let's dive into the results. So one of our initial areas of exploration was to explore the impact of different site harmonization strategies on model performance. But before we look at the findings, let's figure out what site harmonization is and why it's necessary. So imagine we're collecting data from different locations, each using different scanners, like a Philips scanner at UBC Hospital and a Siemens scanner at Vancouver General Hospital. These varying scanners and locations may introduce inconsistencies in our data. So site harmonization addresses this by standardizing the imaging data across different sites and scanners. And the site harmonization process involves applying correction techniques to reduce the variations in image quality, acquisition protocols, and scanner differences. So the aim is to minimize the confounding factors from site-specific differences and to make the data more comparable and the analysis more reliable. And in this study, we compared 7 site harmonization strategies. There are no site harmonization, GLM with site, compat GAM, compat age preserved, compat, SMA, and ICVAE. So these are unfamiliar terms, but they all just different site harmonization strategies. And for evaluation, we used the 2 key metrics. The one is mean absolute error, MAE, and the other is correlation. So our goal was to identify a strategy that yields a high correlation and a low MAE. But surprisingly, the result from the figure, as we can see, showed that the strategy with no site harmonization actually has the best performance because it achieved the lowest MAE and the highest correlation across different age intervals. The next, we turned our focus to understand how age and sample composition affect the model performance. So we split our discovery sample into different age groups, like every 10, 20, 30, 40 years, and the entire lifespan as well. But interestingly, we observed that as we increase the span of each age interval, the correlation across these intervals actually generally increased. However, the MAE doesn't follow this pattern. So the tightest interval here, the 10-year range showed the lowest MAE, and the 40-year interval were the next. So to consider the balance between the high correlation and the low MAE, we conclude that dividing the sample into 2 age intervals, specifically from age of 5 to 40 and 40 to 90, will provide the best model performance. And this approach provides the right balance to ensure our model is both precise and reliable across a broader age spectrum. The next, we explored the necessary sample size required to get a stable brain age model. So despite our initial large data set, which includes over 35,000 healthy individuals, we questioned that whether such a vast number of participants is truly needed to develop a reliable brain age model. And to explore this, the discovery sample was randomly partitioned into 30 subsets and ranging from 200 to 6,000 participants in increments of 200 without replacement. And the data demonstrated that both MAE and correlation did not stabilize until the sample size exceeds the approximately 1,600 individuals. So from this analysis, we conclude that a minimum of 1,600 participants is crucial for developing a stable and accurate brain age model. And this finding provides a guideline for future research in terms of the scale of data collection required. And finally, we evaluated the longitudinal consistency of our models. So this involved using the models on brain scans taken from the same participants at 2 separate time points. The analysis focused on how well the models perform over time across different age intervals. And we discovered that the models divided into 2 specific age bands, 5 to 40 and 40 to 90, demonstrated the highest consistency in longitudinal data. Because we found the 2 age band model showed the smallest variation in MAE and the correlation between the 2 time points. So essentially, the differences between the MAE and stability and correlation provides the strong stability of brain age estimates over time. And this robustness is crucial for ensuring that our models can reliably track and predict brain aging, even as the same individuals has changes over several years. To summarize the finding, the model segmented into two age bins, which utilizes no side harmonization strategy, will be the most effective one. And we also demonstrated that when applied to a sample size exceeding 1,600 individuals, the model not only has the optimal performance, but also maintains its robustness in both cross-sectional and longitudinal data. And to provide better access to our model, we've made it available through an online calculator on the Centaur-Brain website, where a user can upload their own structural MRI data and to get the results. And the Centaur-Brain is just a platform dedicated to the open science and web-based individualized neuroimaging metrics. And additionally, the manuscript is now published in bioRxiv, and it's under review in Human Brain Mapping Journal. So for more information, please, you can scan the QR codes to learn more. And I would like to give special thanks to Dr. Sophia Frangu and Dr. Rayang for their invaluable guidance in this research. And thank you to all the collaborators involved. Thank you very much. And yeah. Yeah.
Oh, is there like a microphone? Okay, yeah, whatever. So thanks. I just have a conceptual question about this. So what you're working on here is making the model fit the data as well as possible, like, based on how closely it matches the chronological age, correct?
Yes.
So that seems sort of at odds, though, with the idea that deviations from the model have a biological interpretation, like if someone is aging, their brain age is off from what the model predicts. Because I can make a model that fits the data perfectly, and there be no deviations, or I could make a model that fits the data really badly, and then there's lots of deviations. How do you know what the right model is if you're going to look at the residuals and say they're significant for some interpretation of the person's health state or something? Do you understand the conceptual?
So, yeah. So the idea of the study is to build a model that can accurately predict the brain age, the biological age of the healthy individuals. But apparently, most people won't have the exactly same chronological age against their biological age. So more or less,
that's not just noise in some sense versus something biologically interesting. How do you evaluate that?
Well, so first of all, the training set, the discovery sample we used has over 35,000 healthy individuals, and which is a massive data set. And they all healthy individuals, which means that in the ideal case that there won't be too much difference between their biological age and the chronological age. And we built our brain age model based on this huge data set. As I mentioned earlier, we used the MAE and the correlation to evaluate the accuracy and the performance of the R model. And the thing is, to get an accurate model, the MAE should be low and the correlation is high. And in terms of like you mentioned, the residual, healthy people more or less has different chronological age against their brain age. And what we focused on is the brain age, which with the capital letters AGE, brain age gap estimate. And this term can be interpreted in many ways, especially in the context of the neurological and psychiatric disorders. And this metric is very important because sometimes people can get some insights out of it. So for healthy people, they age they don't age too fast or too slow. They will have the same brain age, biological age and chronological age, but otherwise they will have a huge difference. And this work is just to have the brain age model that can accurately predict their biological age. Yeah, that's it. I hope it does address your question. Okay. Yeah, thank you very much.
Next, I'd like to welcome Dr. Sabrina Leslie. She earned her PhD in optical physics at UC Berkeley and followed it up with a postdoc in biophysics at Harvard, where she invented convex lens-induced confinement, which established her as a pioneer in single molecule investigations. Recently, she was honored with the Young Investigator Award from the Biophysical Society of Canada and was elected to the Royal Society of Canada College of New Artists, Scholars, and Scientists. Her team is currently working on developing single molecule and single cell imaging and analysis tools, as well as mathematical models for molecular interactions.
That's where it's showing.
Yeah, Oops. I don't know. I don't know.
I think that just worked.
No, it's not showing on the projector. Oh, there. Okay, I think we're good now.
And then there's a way to make this disappear. Fluorine meeting. All right, is that good? Okay, yeah, so thank you very much for the introduction, and it's great to connect with the community here. I guess this talk is also in some conceptual way timed with my arrival and arrival of my lab to the Michael Smith labs in Vancouver. And yeah, I'm very grateful for the introduction, the ability to connect with you and people on Zoom. And thank you for the very kind words. So as was shared, I did my studies in Berkeley after first beginning my undergraduate math and physics training here at UBC. So physicists love conserved processes, and there's no place like home. And yeah, it was shared with me there'd be many bioinformatics and students and colleagues in the audience today, and I might begin as a tradition of Van Buck, which occurs also later during the pizza and beer with just a bit of a personal story. But I did make that shift from the very fundamental physics, optics development to, as a grad student, soul searching, really wanting to apply that to living systems. And then built a first lab of my own at McGill. And then during the lockdown, I see some of my team members in the audience, somehow moved here with our lab and rebuilt. So I think maybe it'll be this fall where we have our, full first batch of papers out, lab fully operating. Quite a bit more expansive than before, and really an honor to be able to pursue this dream that I'm going to tell you about today. Yeah, and just to connect a bit more with the students, I mean, especially those who are in undergrad and grad school, I totally get what that's like, figuring out what you want to do. But I started in honors math and physics, which is kind of like a theorist training program. Very intense mathematics related to the bioinformatics people are doing here, in a sense, focusing on theory, modeling, teasing out the data. But in the summers, I had the chance to work in some labs, and I was very fascinated by imaging and seeing what one couldn't see before. And in my PhD, the ability to kind of design experiments, I've been learning about quantum mechanics and cold atoms and control, and also do the theory was really appealing. Especially many parts of physics get spread out. But at some point, you know, although I did learn to build instrumentation, optics, control fields to trap single molecules and atoms, lasers, it was sort of starting to feel a bit remote. The conditions I was looking at these molecules in were fuller than deep space. So on a lot of reflection in the later years of my PhD, I really decided to take a leap in my postdoc and shift, instead of from the fundamental physics questions, maybe things like, how is the sky blue? Or, you know, is there life on other planets? Maybe that's not physics, but you use astronomy to search for that. But instead, kind of look inside, you know, what are the rules of the molecules of life? How do we stay alive? How is DNA repaired? Structure of these biomolecules, just kind of maybe the physics piece of the biochemistry, regulating that. It's just amazing. And the inspiration of the group here, we will have built 10 different microscopes at different sort of scales, very complementary to the work of health science developers, really all at that most granular scale, just dividing it up. I mean, it's to look at single molecules, not in ultra high vacuum, but instead, you know, molecules of life, DNA, RNA, protein, in the wet, squishy interior of the cell, which is crowded, confined, and really complicated. And, you know, what does that mean, and how do we fit? So, you know, our collaborators might develop a lipid RNA product or a drug, and their measurement of, say, on and off rate or affinity would be done in a test tube. And that assumes all the molecules are the same at some level. Or, you know, other techniques really start to get at single molecules, but an obvious way to first look at single molecules is to tether them, hold them in one place. So surface plasmon resonance, you sort of attach one species to the surface, flow reagents over. And many folks in the audience have probably looked at that kind of data, complementary to genomic cell knockdown data. And it can be a bit artificial because, as you know, these RNA, DNA, proteins are complicated, they have structure. So tethering them near a surface is complex in a number of ways. So the general imaging concept I developed, and thanks again for the kind introduction, was to kind of get away from all that. And maybe it's a bit ironic that I was so thoroughly trained in all this optics, electronics, etc. And what I wanted to do in moving into the field, I was just learning new methods, is to actually remove all the variability from my experiments and simplify. So in learning to pipette and use a fluorescence microscope, initially by just putting a lens on a surface, do something like this. So what you're seeing here is actually the same kind of imaging framing we use today. You're looking up into the objective of a microscope, and the flow chamber is such that it's quite thick when you first look in. I mean, it's thinner than your hair, but 30 microns thick, let's say that's thick. And in it, we flow biomolecules. These are lambda-phage DNA that are stained. And then we kind of mechanically squeeze so that the molecules are just floating between two thin sheets of glass. So you might ask, who would bend glass over a $10,000 or $20,000 objective on a super fancy fluorescence microscope? I guess that was me. But it makes sense, right? That glass is very smooth, doesn't absorb. You can get nanoscale control. Very hard to tilt things on the nanoscale to be perfect. Better to have something round and flat and bring them together and look on such a small scale that they're basically effectively flat. And so then sort of, if you imagine looking inside the test tube, in this particular example, we're also stretching out DNA maybe to inspire the many genomic applications at a single molecule level that folks in this audience might immediately think of. But we're, in such a way as to not tether, separate out the molecules, look at them, and we can control their shape as an additional nuance with patterning shapes on the surface. So this was all initially included in a platform called convex lens-induced confinement, because effectively, geometry, you learn that if a surface has any radius of curvature, I guess even if it's 0, and you bring them in nanoscale contact, then you have a lens on the surface. So that's the concept. The implementation that we worked away at, that it would be better than, me pushing something with a rod in my finger, is just in starting at McGill, flexing a piezo to get that nanoscale control. And so this is one implementation of Click, which works quite well with a little instrument sitting in the lab, where we're looking up, the samples flown in between the 2 glass sheets, and this little pencil-shaped object with an upside down lens at the tip called the Click lens is flexed down. And today, I see on the engineering side of our group, as was mentioned, I'm in physics, but also affiliated with engineering, the genome science and bioinformatics programs here. Is we do like to refine instruments, we like to build. And this particular instrument is the one that my students are using, where we have a little tube, the convex lens at the tip. We squeeze molecules into this thin imaging region that we're looking up into. And it just sits on top of an inverted, pretty fancy microscope we have from Nikon, but would mount to any inverted microscope. So if you're curious, before we get into today's focus topic, on a bit more of the background, a bit more technical, it was actually very fascinating in the beginning to explore some of the conformational physics of DNA. Where I wanted to head is how does that impact interactions, and being able to eventually look at molecular search, live cells, and in controlled solutions back to back, as well as also delivery. But science is very one step at a time, and it takes a lot of time, very deep, rigorous work. So our first publications both served as a deep study of the confinement physics that we could access, but also as a refinement of the tool itself, because the theory we were comparing our experiments to was so rigorous that we had to really study our own tool. But we had some early articles in NES and macromolecules, looking at confinement physics and different geometries with applications to genomics. We've been curious about the impact of confinement on chemistry. There's a cute example of encouraging ligation of constructs in nano rings compared to bulk. And then we've gradually shifted to really looking at structured DNA and interactions of DNA and proteins with target sites, where now we're getting away from influencing the structure, but in those more native-like conditions with these early papers, and also more recent publications in nucleic acids research by our graduate students. So we're very excited about all the places we could go scientifically, and our motivation is really to get to that next level of detail. Look at one molecule at a time, follow their multi-step kinetics and their variants. So this is something that's obviously emerged in the therapeutic arena, especially around structured RNA targeting therapeutics and lipid particles carrying those RNA. Very complicated biological objects that we really need to study in detail. And we have met that original goal of the single molecule studies closer to life, but also removed in some way. And our work, of course, occurs in a broader context. Those of you may know that super resolution and single molecule resolution was awarded to physicists in chemistry departments, Nobel Prize in Chemistry in 2015, and then more recently, optical tweezers, a method for trapping particles and molecules. And so these kinds of methods are, I think, appreciated by the scientific community more and more, even though abstract at one molecular level, whereas biology occurs in such complex environments, it stands to reason that those insights will help with development. And perhaps what I'll show today is an approach where we really try to excel at single molecule data with high throughput, without the tethers and practical impact. It's kind of our inspiration. And one of the other things, I mean, in the cell, you have structured molecules, drugs binding to targets or delivering lipid particles, talk about today. And things are really out of equilibrium. It's another key thing. So it's not good enough for us to look in a test tube or tether species down. You're going to see how using confinement physics, we can entropically hold these lipid particles, exchange buffers, cause them to release. And earlier on, this is an example of a DNA compacting when changing the reagent. So overall, our research program, in the first bit of my talk, just to introduce myself and lead up to our feature application today, is enabling us, and for students in the audience, I think it's already discussed coming in, we have some very nice papers in nucleic acids research on structured DNA and oligo enzymes interacting with those. The new technology development that we've been pushing the limits of studying these interactions is not yet published, but has been presented in a few conferences where we're looking at short anti-sense oligo binding to RNA targets, and their that last event of the drug target hybridization as a function of modifications and structure competitors. That's sort of underway. And then the topic of today is actually how do you deliver these particles, and taking a closer look at the delivery agents themselves, the lipids that are used to carry these RNA therapy cargo. So sort of the last little bit of my intro before I shift to our story about the lipid RNA particles. Our dream is that, yeah, we could characterize in bulk these therapeutics in collaboration with health scientists and get at those last moments of hybridization and cleavage, for example, of an ASO, and take very high throughput scan of them in our instruments. And one little last bit of the intro, and this will come up again today. I'm looking at the lipid RNA particles, where we can look not just in one fluorescent color, but in two, or actually we've set up 4 now in some of the microscopes upstairs. So we can see, for example, drug target, hybridization, also cleavage by an enzyme, and other partner proteins mediating those interactions. Okay, so that's kind of the big picture, everything from where I came from to where we're headed. And if you're curious to visit the labs, you're of course welcome. So today, we got into genomic medicines, and coming from our background in DNA biophysics and optics. And my first sabbatical, where I went out and chatted with scientists in oligotherapeutics field, which is a smaller field, but just so cool that one could develop therapeutics out of RNA, DNA, and see if this might be of use. So this is before the pandemic took off, and therefore the field of lipid RNA medicines went from obscure to household name. But what we started doing, and we've continued to do, largely in collaboration with Peter Cullis here, I guess since 2018, is to use our approach to take a very detailed look at these fascinating lipid RNA constructs. So the idea is that, they are really quite heterogeneous. They've been injected in billions of arms in all of us. They interact differently with all of us. There's a lot to learn. A lot of their properties are not standardized. So there's also this aspect of a practical contribution, like thought leaders at NIST or measurement. Could we, in our tether-free single molecule imaging platform, provide a quantitative readouts of the size of every lipid particle, its RNA cargo, maybe how they interact with each other, study, we all read the press of storage in different conditions and why is that necessary? Following on those things with exquisite detail. So Albert Kamanzi and strong team have applied the click imaging since about 2018. Their first paper is in ACS Nano 2021, a commentary or 2 in 2023, another one in final stage of review right now that'll hopefully be out soon, and several follow-ons where I am going to share in this talk data on the mRNA lipid particles and parallel work in cells. So I guess this is a system where we're taking first steps to realize this dream of the very detailed single particle data all the way up to the journey inside cells and be able to correlate properties. But I do want to emphasize that there are a lot of other applications that we're also pursuing on a longer time frame in the lab, which sort of include this kind of vision. And I'm speaking broadly also because of the bioinformatics audience, and perhaps this being a sort of additional data set you didn't know about, or open questions at the single molecule level, where we might be able to collaborate here through the Microsmith Lab. So in this particular look, following the theme of this talk of lipid RNA particles, we're loading, like I showed in the video, the lipid RNA complexes in the flow cell, deforming the roof so that they're individually trapped and diffusing in wells. And we do use a little bit of fluorescent stain on the particles. These are stained in one channel so that we can trap each of their trajectories. And then by developing the theory of their diffusion in confinement and very quantitative simulations to emulate the data on artificial samples and then control samples, we can extract for every particle the diffusion in confinement. And if you look at the supporting information of this paper, really several years of work, it could have been its own paper, but really making sure that those diffusion measurements are correct, which are one measure of particles' size through its mobility. Now, beyond that, we're obviously getting multiple bits of information on the same particles. And the way we're kind of thinking about this conceptually is that they have different sizes, different shapes, different payloads. So perhaps our data is a kind of fingerprint of these formulations. So the first control experiments we've done with back-to-back cryo-EM, done by the folks in Natalie and Peter's labs over in LSI, is at 2 different pHs. We've collected the single particle tracking data with the log of their diffusion or converted to size on the x-axis, and the log of their intensity on the y-axis. So if this was like a completely uniform sample, it would be one cluster. If it is the case that the samples have a surface area that they do, and the dye, the stain, which is giving us our readout here, is spread out over the surface uniformly over many, many, many thousands of particles that are required, you would expect on a log-log curve for the slope to be 2. And then that does mean there are outliers for you to detect the slope, and there are. So we kind of capture the heterogeneity of the sample, which is important to understand, and we're going to correlate that to the uptake and entry and dynamics in the cell. We can characterize that also from the perspective of storage and aggregation. And we see there's sort of a trend here with the complexity, but that's not so trivial. These are not like our cold atoms in ultrahigh vacuum. And then when we change the pH, and the reason this is quite interesting is, and you may know that these lipid RNA complexes are, at least at the moment, formed typically in acidic conditions, the prior example, and then shifted to neutral conditions. And these were prepared in Peter Koh's nanomedicine lab. But when they are prepared and stored in neutral condition, this distribution, or I'm using the kind of idea of a fingerprint, it's a complex graph to sort of communicate, is actually quite different. First of all, the slope goes from 2 to somewhere between 2 and 3, here 2.3. And the distribution is not uniform. It's really quite heterogeneous. You can see by cryo-EM that the particles are sometimes in pairs, triplets, and they do have more layers. So the fact that the dye is interpolating into the volume, a uniform volume stain will give a slope of 3, and a surface stain a slope of 2. It makes sense that we got something between the two. So if one did want to contribute precise measurements, for understanding the biophysics of interactions and looking for impacts, but also possibly for quality control or helping standardize across different labs, that's important. And then the next thing we needed to do, which we actually couldn't do right away, is we wanted to really count the number of RNA fluorescein per particle. So we do this using something that's usually a nuisance in fluorescence microscopy called photobleaching, where the dyes on the particles will actually kind of bleach off. So inside, this is not a simple thing. There's a very complex fluorophore, and it's interacting with light, and it can kind of absorb and emit photons enough times until it's actually modified, often by interacting with oxygen, so that its spectra is such that it doesn't emit those fluorophores anymore. So, but anyway, in practice, one does this at higher intensity. So we learned to do sort of very long tracks to get these nice trajectories, pull up the diffusion and intensity distribution, and then very rapidly run with a higher laser, bleach off the cargo, and therefore count the number of RNA per particle. So this is a way, in principle, with high throughput, one could run in an automated fashion for a flow cell, hundreds of formulations a week to get this very detailed information missing in the field. And I was surprised there were many developers coming to us, at least half a dozen major pharmaceutical companies the past year or two, who are they don't actually know how many RNA per particle they have. So the analytics in the test tube, you can imagine it's complex, right? Because you mix a certain amount of reagents, you have a bulk estimate of the RNA and the particles, but you might not really know how many RNA are in there. So this has been of great sort of practical interest, which is, I guess, one of the applied aspects of where we could help make, a contribution back to society. One of the outputs of this first study we did, so the very first lipid siRNA, so the first lipid RNA, but it was an siRNA silencing therapy, for those familiar with that, that was released. That was by a company called Alnylam in Boston around 2018, 2019. So that actually positioned the lipid particle carrier as a very good prospect for the mRNA vaccine when the pandemic hit, for those who may not know the history. But at the time, the size, payload, and structure had not yet been looked at in 2018 in my first sabbatical. So Peter Collis's group did this cryo-EM work, and we back at McGill at the time ran many copies of the sample, only to not find on the same scale this nice correlation. We have this uncorrelated nugget we talked about in the beginning. So if you look, we have many thousands of dots here, because it's really quite surprising that on the same scale as before, we don't see those outliers. And it turned out in cryo, the data that we saw later, and maybe this is important for sort of blind data acquisition, that the RNA was really nuggeting up in the particle, which is consistent with the random distribution. So if you've had no correlation, a large nugget of RNA in a small particle or a small nugget of RNA in a large particle, that makes sense. So then with setting up the methodology and deeply understanding its limits, more recently, and this part is not quite published yet, the level of rigor and acquisition in a study like this is a bit intense. We've shifted our attention to the mRNA. So now we get to, I guess, the title of this talk, a detailed quantitation of the mRNA lipid particles, and starting to vary their fundamental properties and relate to their journey in cells. So this is kind of the big picture vision of the lab, but for a very specific and clinically important class of therapies. And this kind of data, maybe it would fold into some of the bioinformatics and rational design work being done on a larger scale. So we've built a 2-color imaging system here where we can track separately, for example, if there's an empty particle with no mRNA, and this is after developing labels where the impact of the mRNA is not seen both in stability assays and cryo-EM, you would see just a slowly diffusing mRNA lipid complex in just one channel. Whereas if the particle did contain an mRNA, one could track in 2 channels that construct. So particle by particle, one can extract the intensity and size, and thereby look at the correlation and do cryo-EM. These quite fascinating structures whose biophysical details are not fully understood. So recently in the lab, and I'll emphasize this is data in progress, we've been curious to look at that, fingerprint of the lipid RNA particle, understand the shifts in the full versus empty particles. Perhaps there's also an opportunity to eliminate the empty particles at the scale of other work or questions being asked about toxicity. Are those many empty particles important? There's also been some work that has been published in the past couple of years citing quite a high fraction of empty particles, which sort of disturbs some developers. So one could imagine. So it's been important to kind of come in and do these very quantitative studies. So I will emphasize that this is the formulation we started with. And then we're varying the ionizable cationic lipid. So the one that was first introduced in the MC3 formulation in the Onpattro therapy, it's a specific lipid that was developed to get these sort of lipid particles to work, deliver the siRNA. That was commercialized by Acuitas and Alnylam. For those who know the great success story, one the spin out to UBC, Acuitas developed that. And then when the pandemic hit, I guess both Moderna and Acuitas and Pfizer and BioNTech were ready with these slightly different ionizable cationic lipids to carry the mRNA. So here we're kind of permuting the type of ionizable cationic lipid and looking and seeing its fingerprints. So at in one case, you see the fraction of mRNA loaded lipid particles versus empty ones in red and blue. And the red dots here are overlaid on the blue, so you're not seeing the entire blue cluster. Is fairly even. Over here, we have quite a higher fraction of loaded versus unloaded. And their other significant finding is that there is a shift in radius. So the larger particles that are full of mRNA, where the number of polymers is digital, is actually quite significant difference in size that we're observing. So just for this particular trial data set, and I want to emphasize this is being done fairly rigorously now, we do see quite a significant difference when we vary the MC3 versus SM102 versus ACL0315, different limits. And I could get into more of the biochemistry of that. I think it's not unreasonable because the mRNA here is quite a large polymer. So in solution, its radius of gyration would be not too different from the lipid particle itself. So when they're mixed, the idea that some are full, some are empty is not unreasonable. But the fascinating thing, and this has been published elsewhere, there's a postdoc and incoming assistant faculty of UBC who's done some detailed work in cryo-EM as well from the Colas lab. You see that in cryo-EM, the RNA there is hypothesized to be this little pocket sticking out, like some call it an acorn, our collaborators in Sweden. Here it's called a bleb. But the phase transition, how the RNA is compartmentalized in that particle, whether it's kind of showing up as a shell or a series of rings in cryo-EM, or a kind of acorn or bleb at the surface of the particle. This has been quite an interesting outcome. It seems to depend a lot on the surrounding solution, the type of lipid. It also seems to depend on how the particles form. So I think over the next years, this is one thing where some hypotheses have, pointed to this having a very strong connection to efficacy. Other folks have told us that they've seen these for years and they don't matter. So we find that, a little peculiar. So what we'd love to do is systematically check out and understand the biophysics of RNA compartmentalization in the lipids. Many people looking at this in different ways. What is their distribution of payload? What is their size? How do they move? And then as they enter the cell, I think it's going to get really fascinating as they interact with the endosome, how they're released. Do these synthetic lipids, matter for ultimate expression? Do they interfere with activity at the ribosome? It's very neat, very complex material. So we've, of course, varied parameters of interest. You've heard in the news the amount of PEG could be a real thing to almost take out of the particle design if one could. It has to do with personalized allergic reactions to these things, at least from the media. I suppose that over the many years, systematic studies of folks' responses to these mRNA lipid therapies and the bioinformatics of those might be a big contribution of audience members here. But here, at least in our single molecule microscope, we're seeing, as we vary the PEG, very large shift in shape and size. PEG allows stability and controls size. So it does make sense if you push the system to be a little bit smaller, that there'd be less RNA per particle, so more empty versus full particles. But the impact on these cryo-EM acorn and bleb shapes is also quite interesting. It'd be nice to work with a soft matter mathematician, theorists to deeply model the formation and structure of these particles, but this data is at least showing us what kind of structures they can take, and as we vary the different ionizable lipids with the same PEG, or the different PEG with the different ionizable lipids, again, we see quite large variations in payload and structure. So I don't want to go through too many of these in this talk, but I wanted to give you a feeling that this kind of data, quantitative, could be used, back to back with bulk characterization, and it could biophysically matter with delivery. So that's, yeah, and building up the lab since we moved, we've been able to finish and publish our assay over the pandemic. Some of this data has been taken during lockdown for the first paper, and then the mRNA lipid particle characterization here at MSL. We've also been very fortunate to receive CFI and Pacific Hand funding, which have allowed us to begin to work towards a stream of not only characterizing single particles, their diffusion, their motion, and size, their interactions, but also their journey in cells. But as a baby step, I told you about that reagent exchange and how we're very curious about it. We're just dissecting this problem. We got a little curious and distracted, maybe. But this is also an important problem in its own right, and just how the particles fuse to each other, and then eventually how they fuse to the different compartments of the cell, the membrane of the cell. So this paper is hopefully about to be accepted, and I have presented this data before at international conferences. And it's a study of being able to kind of hold those particles without tethers while exchanging reagents and look. And this is of practical importance because actually, as we talked about earlier in my presentation, we do vary the buffer from acidic to more neutral conditions in manufacturing. So one's curious about the lipid particle reactions, one can get a trace. And here we're using an optical technique called fret, where we have half these particles in red, half in green. As they mix and aggregate, those dyes become close together, and we can actually get red fluorescence out of green dyes when they're close enough together. Like if you hit a bell and had another bell nearby, you'd hear the second tone, kind of like that, but on a 6 nanometer scale on optical resonance transfer. But we can analyze these particle-particle reactions as they go and choose the concentration. And then this is a new kind of data. So when we want to answer these questions, do they fuse at all? Imagining in our minds the liquid scrambling, how does this depend on pH, salt concentration? And it's a nice model for also other systems we're starting to look at. One can imagine taking a rigorous approach in parameter space, so varying separately the ionic strength, or a combination of pH and ionic strength. In cryo-EM, we again look at the structures at the start and the end of the dynamic process, and then we actually fit each of those curves, and we decided to pull out the maximum saturation intensity and a rate constant, and in a log-log space, because we discovered these rates are very, very different as a function of formulation. We see the slowest reaction on the red arrow here. We just vary the ionic strength. The fastest, we actually use the most practical. This is what's used in practice when the particles are manufactured, the pH is dropped or raised from acidic to neutral condition. And then to tease and study the role of pH versus salt, we varied conditions with few permutations. So in the end, we see that pH really drives the process. When we're varying the pH, it goes much more quickly, and the saturation level can be different. And also these little structures and how the RNA is structured inside the particles, we notice is quite different each time. So it'll be nice to work towards a generalized model of this. We also varied how much RNA per particle is seen. So you see these ring-like structures here. And similarly, when just the salt is varied versus the pH is driven, there's quite a significance in the rates. And PEG is one of the things we're also just really curious about because it regulates size, but higher PEG slowed down the fusion activity as well. So lower PEG allow things to go fast, but they're also more unstable. Yeah, so that's another piece of work that we've been focused on the past couple of years here at the MSL. And as we move forward, there's a lot of questions about affinities of different proteins to the lipid particles, how they survive in plasma, different tissues, with targeting, you know, which proteins accrue on the particles and how those interact with different types of cells. So we're hoping in the long term that this will allow us to explore those questions very quantitatively and back to back with screening data on single cells. So in the last few moments before the questions, I wanted to share what we're doing there. So the idea, and going back to the start of my talk, is we'd like a tether-free platform for looking at range of biomolecular interactions, see dynamics in real time without tethering them down, create those increasingly cell-like environments that we hardly understand by adding crowding, confinement, etc. But then perhaps most realistically, you know, also following those observations inside the uncontrolled cellular environment. So we're doing our best, and to that end, being able to look at the fusion against the particles, the model endosome, the membrane, all those things, looking for correlation is a big effort in the coming years. And then following the particles against the live cells is really quite important. And if members of this audience aren't familiar, so there's a challenge in the field. If you look at the literature, it's cited that, you know, if you put in 100 particles, 1 or 2 will do something for you. The efficiency is less than 1 or 2%. Now, I guess it'll be remain to explore exactly what that means, but from a single particle perspective, we'd really like to rigorously examine this and, you know, correlate to the particle properties themselves by following the individual particles as they enter cells, looking at their co-localization with the endosome, and just simply counting numbers like we're doing with the lipid and RNA themselves, and bring some rigor to these statements, and in conjunction with situations where the bulk data already exists. So that's where our partnerships are important. So that effort has been begun. So now we need more kinds of microscopes that we set up. So earlier in this talk, I talked about confocal microscopy and the very rapid single molecule wide field imaging, and then a slightly slower lipid particle wide field imaging in the main body. And now we're going to reintroduce confocal, but this is scanning confocal microscopy, so that we essentially don't cook the cell. The scanning allows us to trace all the planes of the cell in 4 different colors. So the idea is that for a particle we've very precisely characterized, we could lock onto it, scan around it, and follow its journey into the cell. So we've received the scanning confocal microscopes fairly recently and learned to use them. And one of the nice things is one can stay in the outside of the cell, the nucleus, in different colors, also the RNA, the particle. You know, there are markers, biochemical markers previously developed for endosome at earlier in the stage where the particles can get trapped. So that's been a very large effort. And then given we're in the, you know, Michael Smith labs, and very dreaming big, it would be so cool if not only could we get those nice dynamic trajectories of every particle in the cell, but also connect with the bioinformatics, genomics, and proteomics field, like the kind of data we've seen earlier in the use handling, where well by well, we could sort of zap the wells, release the cells, and sequence the individuals. Now that turns out to be kind of expensive in terms of all the sequencing assays, but there are assays that can work. And our friends And the instrumentation group at the BCCA and genomics and proteomics here are quite excited as well. So we're hoping that that's something we'll achieve. And it is different than looking at flow cytometry or an ensemble of cells. And of course, that's something we'll have to compare, because you'll know the complete kind of history dynamically of therapy or delivery and its omic outputs. And that's different from looking at, say, an ensemble with single particle resolution, because the level of information is deeper. So for example, if there are different populations of cells that responded very differently to the same drug, and they had different phenotypic properties, and the drug's journey was also different, we would see those clusters in terms of their behavior in time and sequence outputs. And that's different from just an ensemble average, because you'd have the correlation between the two and information that previously wasn't known. And of course, single cells have been correlating well in some therapeutic development, but a lot of rigor is required to establish that correlation and show this is relevant. But it does stand to reason that the last event the drug is against its target and in its cell, this kind of data with the right design and controls of experiments should eventually be quite powerful. So here's what these kind of pictures look like. So we're learning the analysis is quite complicated. It took us a while to learn to track particles in wells and compare that to artificially generated data, but the cellular data with the fluorescent labels and also without is even more complex. One has to reach to deep learning and combinations of both mechanistically understood as well as just machine learning and deep learning, where the sort of entirely different approach to data analysis are used. But on the same scanning confocal microscope, with help from our collaborators, so both in Sweden and the Department of Chemistry here, we can also scatter light. And I guess this kind of, in terms of the story, goes back to my PhD. I had shared at the start how PhD was in imaging these cold atoms in ultra-high vacuum, but we actually used phase sensitive measurements, and the whole field, if you look at atomic physics, has moved from fluorescence to phase. So the nice thing about scattering light and interferometric measurements is you kind of see scattering off everything. The signal is more messy and complicated, but you can learn a lot about structure, and overlaying on that, we can fluorescently tag the lipid and the particles, say in green and red here, put in stains for endosome at different stage. And so over time, be able to get the maximum amount of information as a function of different cell types and permutations. So that's the work that's kind of ongoing right now. Yeah, so I think I've supposed to allow some time for questions. So I finished 10 minutes or 15 minutes to spare before the pizza. And just to summarize, so from this talk, I guess we've introduced a new way from the basics up for trapping and following single molecules and particles in conditions that approach cell-like conditions. The particular application to lipid RNA particles has allowed us to see they're really quite heterogeneous, and detail about that correlation between size and structure and payload. Their fusion kinetics are complex, and the next steps are to look at fusion against the membrane and artificial endosome and control conditions. And the dynamics in cells are also incredibly complex, but with the resolution we have, we can follow single particles in cells. And the next step is to quantitate as a function of the same variations in formulation, uptake, colocalization, and eventual release of the RNA. And to that effect, we have built a number of microscopes. I showed earlier in the talk, kind of at the single molecule level, single particle level, and cell level, requiring advanced technology and confocal, wide field multicolor fluorescence, and then scanning confocal for the cells. On top of that, we have set up the system with robotics, because we do understand to connect with bioinformatics data, and perhaps these would be new data sets kind of folded in at the most microscopic level. The expectations are to run, hundreds or thousands of samples a week, not 4 or 5. So we're working with support from BCCA, and we've also spun out a company to help play the role of bringing this to practice, as was mentioned at the start of the talk, so that this kind of data can help develop medicines, not only in the university context, but also in the hands of developers who can move at a much faster pace sometimes, or just a wider variety of work in the industry and sectors around the world. So yeah, from the early honors math physics to building up this lab, I guess I'd like to open the floor to questions, and especially thank my team. Yeah, over the years, there have been a number of scientists who have contributed, and today we have two MyTex postdocs, Albert and Ari, as well as the visiting postdoc from Sweden, Eric Olsen, who's in my lab, come across the great ocean to come with us, which is great, and Cindy Shaheen, who was actually first graduate of my lab from UBC and first woman to earn her PhD in my lab, so that's quite heroic. In physics, we're working very hard on increasing diversity, as you'll see, but the key is to support and encourage students to continue in their careers as scientists. And then we have a number 6 graduate students who have contributed. The ones in green focus closer on this project, but in our group, because the imaging platform is so collaborative and central to all the projects, there's actually like a lot of interplay between projects. So I think everyone's contributed something. And yeah, well, a number of collaborators. I think Peter Collis's name and his team came up many times, as well as in Sweden, had a lot of help for the surface chemistry and the complementary measurements on the surface. Have a longer history, but a lot of really important insights that have guided some of the practical challenges of coatings and working with our devices. So yeah, today's a great chance to connect. And if you are curious about any applications, I tried to keep this broad enough and yet specific enough that if you're thinking of other applications or projects that you might like to collaborate with us on, just knowing this kind of platform exists, feel free to talk to me today or just come visit the labs or the students at any time. We're in the NCE 323 area where Carl Hansen used to be, and the MSL 269 area where Robin Turner used to be. He had quite an amazing optics space there, so quite beautiful for these single cell and ICF microscopes to be set up. So we're very grateful for that, and of course grateful for your attention, for coming out in May at a challenging end of year time and beginning of summer time. Thank you. Any questions, especially from the students?
Oh, I have a question actually. So when you're doing, you said that now that you're looking at single cells, you're using deep learning, is that correct?
That's one of the approaches to deal with to look at the ISCAT data, because it's really quite challenging to otherwise interpret.
Yeah, so did you use any of the data that you had from the single particle interactions to build that model? Or was it done from scratch on the
well,
cell data?
It's being built right now, and there are a lot of the publications to look at would be out of there's a group Daniel, which is I'm about to fly to Sweden and meet with Daniel. Better with numbers and pictures than words, but yeah, some of that model has been developed before on beads. I think that's the status right now. So the idea is we co-develop it with, there's like 2 or 3 labs in the world that have ISCAT data of this nature of scanning for full form cells. So the data for them, for example, just hasn't been available at all. But they've been able to begin to do that, I think, with some just polystyrene beads. Yeah, if you're curious about that, feel free to email me. But yeah, as with all of our data, as a general principle, we always train our methods. I mean, the idea is, like you've seen with the beads or the fluorescent particles in wells, With fluorescence, it's fairly straightforward. With iSCAT, we have that data. I didn't show that here. But that also requires more development because it's actually picking out the position in 3 dimensions. And then as those particles or molecules enter cells, because the environment is so complex, it's going to be a challenge to hold on to the local field of view around the molecules and track them, and then interpret things like their diffusion and size. So I would say the state of the art is the iSCAT of beads in solution, and that we will be developing and sort of showing that that is possible with the sensitive microscope and tools. But both due to the nature, I think, of how we do the analysis and the sensitivity of our optics and kind of control of our method. Yeah. So it's by no means done.
No, I just was wondering, like, if you can get from single particles to single cells, and you're using some sort of like AI to build that model, do you ever envision it going all the way to tissues, like without maybe not needing the containment at some point?
Yeah, I think so. I think that's true. I mean, here, I guess we're using the containment for control. So the control is physical for the analytical purpose of pulling out things like size, payload, fusion. Control gives you the ability to make precise measurements. But then in the cells, I guess the confinement is more conceptual if you think about scanning around the particle of choice. Because if you scan the whole cell and show the full image, I anticipate you might lose the particle. It just depends on the timing. We're just getting into that. But let's say it's a conceptual to follow the particle. Once that is working, and it'll be interesting to see how well we can measure the diffusion of the particle and payload. And that's why fluorescence and iSCAT together, I think, are already winning. And we're seeing that already with the particles too. If we have low amounts of stain, lower and lower amounts of stain, iSCAT in the second channel can help train. So that's really nice. So imagine we get there, then yes, because confinement is now conceptual. You had stacks or cells or organoids, you could, if your goal was to follow in this great detail, the journey of the therapeutic entity into the cell, then you could do that because it would just scan in a volume that followed it. So at that point, you would lose all the physical control. But I think that is also where things are headed. Like our collaborators who are interested in looking at therapies that target specific tissues like brain or spleen or what have you, are growing organoids. So I guess the physical confinement is the cell ensemble they grow, if you will, and that's their art. We might just place it on the microscope and scan. So that I think you do go all the way up. And if you want the absolute precision on the same analytes or the same particles or drugs, you do that in the glass walls because you eliminate all the background. So that's the idea. And of course, there may be cases where this is not necessary, where things behave fairly homogeneous as expected. I guess when things are complicated or shelved or that this kind of information can really help overcome hurdles. Also testing hypotheses, like are there, in leukemia has come up with some hypotheses of different classes of bone marrow cells that are resistant and not resistant. I mean, is that true? And then also why? And what can we do to make it better? So that's kind of where we're hoping to make a contribution. But yeah, I absolutely think it'd be lovely to, there's nothing stopping us from putting tissues or organs on this microscope. And we're starting from the bottom going up. So we'll meet our friends. We're more starting from the top, coming down and find, yeah, the best interfaces to work on. Perhaps that's long-winded, but it's good to recognize that a lot of this is really in development right now. And yeah, some of the technology is really accumulation of just decades of work in different biophysics labs. we now are in a position to buy commercial products like the scanning confocal, which we didn't have before. And it is very expensive. But when I think about it, it's probably the contributions of hundreds of peers working and then assembled into this product that we can have. But, you know, to build that ourselves from scratch would also be quite hard. So it's sort of the whole field. Yeah. Yep.
Yeah, I have a few technical questions. For the single particle tracking that was done with Thurs or with wide field?
The data I showed was in a wide field regime. It is true that so turf means,
Could you repeat the question if you want to?
oh, the question was for the single particle tracking, was the measurement done in a, I guess I've tweaked the question a bit, but turf or wide field. So it can be confusing because the objective we have is turf class, but what turf means, total internal reflection, is that, you know, Snell's law, water glass interface, that you've tilted enough that you're totally internally reflecting. Like you just have the evanescent wave at the surface. And wide field is when you're illuminating. So we're in a wide field regime because the thickness of the wells is actually thicker than the depth of evanescent probing near the surface. That would be more like 150 nanometers or so, where it's, yeah, related to the wavelength factor 2 and the refractive index. But yeah, the wells here, this is a technical question. The wells here we chose so that the particles stay in focus when in wide field with that fancy microscope, which is called a turf microscope, with, yeah, the 1.49 NA and 100x or 60x magnification. But we're in the wide field mode because we want that full maximum depth. So we're taking this data in 2D, but these wells are around half a micron thick. The beam is a little bit tilted to avoid back reflections right into the detector, but in the wide field mode. So there's some subtlety, but the simple answer is wide field. And the purpose is to keep that particle in focus the whole time, but, you know, get the optimum geometry for collecting the information. So the depth is the depth of focus of the objective determined sort of empirically for the particular condition. And then the width is to basically have low systematic and statistical error on the tracks, you know, less than 10% for these particles of the sizes that they are. You mentioned you have more technical questions?
Second question. Do you have a plan go beyond laser scanning confocal? Let's see, which spins confocal or loosen. Because laser scanning confocal is okay, the speed is low compared to spinless recommendation thing.
Yeah, perhaps that's a beer and pizza question. But yeah, we could, I mean, we did the estimates of what we needed. So this is all science driven, the estimates of the time resolution and so on. And actually, we did compare the performance, at least in principle, of the Nikon scanning confocal system here, and there are others and that you just listed. And for us, actually, this was the optimal for what we want to do. But yeah, longer term, I think what you're hinting at is, could we go faster and with better signal to noise even? So the dynamics that we're looking at here are faster or on the time frame sort of, or the equipment can go as fast as we need for the dynamics we're following here. Yeah, but there are things we can do that would go even faster. The place in terms of our scientific questions where we want to go faster right now requires better signal to noise than those commercial instruments actually would provide on that time scale. And that's why in the very first example, I didn't spend a lot of time where we're looking at the very rapidly diffusing drug target. Like I showed some ASO RNA diffusing in wells is on a camera. But there I mentioned we're just sort of pushing the limits, and we do use confocal there, and there we go faster. Where, just in case you're curious where we're going, you know, sub-millisecond detection. And the science is, how do the conformational fluctuations of these smaller molecules impact their kinetics? But But for the work in cells, because the motion is slow and slowed further, slower compared to the small molecules because the particles diffuse slower and it goes as 1 over the size, but also slowed further by the viscosity and crowded components in the cell. The microscope we have is able to give us a very strong foothold of what we need. Yeah, but we do imagine we will also build more. But yeah, if you're curious to chat more, we can do that. The other thing we've been able to get recently, and we'll be setting up over the next months, is in terms of pushing the spatial limits, right? So is the storm super resolution system. But that's going to be a new advance, I guess, over the next months or year. Because we're curious about structure. So yeah, next level time, next level space will always be, I guess, pushing it.
So you mentioned for the storm, that is mostly for live, so also big samples.
Yeah, we will have to fix the samples, but where we discover, like, cryo is great, but it's expensive and we have to wait, and it's done in a facility, and if we could cross-train storm, we would be fixing samples like in the cryo images. It'd be pretty amazing to look at those intra-particle details, or maybe just complementary to cryo. Like questions about how the RNA is structured, and then in terms of interactions, how that might interact with the environment. It might relate to potency or efficacy. There's papers and stories of competing different observations for different therapies in the field. So if we could have storm in our lab to very rapidly look back to back with the dynamics, I think it could be interesting on the same samples. So yeah, I think that's where it would come into play. Obviously the fixed cells as well. Yeah, because our dynamic data is always done, say, to optimize a time trajectory or a kinetic interaction. So be at the expense of spatial, where storm fixes things to optimize spatial, but at the expense of time. So our idea is that it would be super great to be able to have, solve the sneaker problem and do everything in one space with all of that information together.
One challenge might be the storm is very strong label, and by reducing label the lipid nanoparticle, the mRNA, would be compatible with the storm?
Yeah, we've looked into that, and it is, but obviously we'll have to do more rigorous checks. But one of the things we do with any dye, and we eliminate a lot of dye and preparation methods this way, is we do check in cryo-EM that structures aren't altered. And also these long time kind of fusion assays I showed have turned out to be another smoking gun, just like is the sample equally stable. Because when you change the charge of it due to a dye, you'll see this kind of aggregation. It's very subtle, but it's a good thing to tell you something's perturbed. And cryo, obviously, if those little structures change, we saw how sensitive they are due to a dye, you see that too. But yeah, we do have reason to believe we're good. We've done some preliminary tests on a colleague's storm and with the dyes that are needed. And there is some work that's very early that's been done by another group that we've talked with. So, but it's right to know that these are challenges. So that's why a lot of these developments take time. Because the details of the dyes and the coatings, it's a lot of work in its own right. Yeah. But as we go, we build up much more expertise and knowledge that will increasingly correlate with good conditions. It maybe won't predict specifically all the answers, but yeah. So one of the things that's been really good too is we're going to be able to solve the sneaker problem further and have the bulk characterizations of this sort of charge and other measurables in our lab, which hopefully will help predict about the dyes. Yeah, but we have some data with different dyes, and it can very easily not be appropriate where you wouldn't expect. Yeah. These are great technical questions, yeah. When I give these talks, we focus on the sort of questions and data inputs and outputs. But in real life, there is a lot of detail and it's very technical. And it relies on a lot of different repositories of knowledge that are not just physics, surface chemistry, yeah, analysis, biochemistry. So that the questions are good. Yeah. Anyone else have any questions today? Or they're all seeking, it's the pizza hour.
Well,
I saw some running back and forth during the talk and only assumed that was pizza related kinetic activity.
yeah. Well, you'll be available during the pizza.
Yeah,
so you-
we marked that I'd stay till 1:30, so happy to join.
So, yeah, so if any audience members want to ask more questions, you can
Yeah.
reach out to Dr. Leslie. Okay.
And thanks for coming. I know it's a challenging time of year with things wrapping up and things starting, so it's sort of a highly fluctuating time.
Yeah.
Yeah. Okay.
I'd like to thank Dr. Leslie for coming.
Thank you.